Assalamualaikum, my name is Karunisha, a final year student, and the title for my final year project is Physicochemical Investigation of Zinc layered hydroxide synthesis for beta-Naphthoxyacetic acid BNOA intercalation. Plant growth regulator is a class of trace organic generated naturally by plant metabolism or synthesized by chemists, serve critical roles in plant germination, development, and reproduction, as well as biotic and abiotic stress. PGR are classified as auxins, gibberellins, cytokinins, abscisic acid, and so forth. Among auxins, beta-Naphthoxyacetic acid (BNOA) is frequently used to stimulate early fruit, regulate stem, root, flower, and fruit growth, and increase the size and color. Auxins has become the first phytohormone or plant hormone found in plants and is involved in a wide variety of developing activities, including growth of cells, differentiation, and patterning of tissues. In terrestrial plants, auxins operates by traveling over short and long distance as well as establishing gradients that preserve cells and tissue polarity through the use of broad array of auxin transporters. Drawbacks of auxins and how to control it. Auxins secreted in fruits and vegetables may be hazardous to human health. As a result of long-term farming, auxins have built up in the soil and are now flowing into the waterways and are equally hazardous. Hence, zinc layered hydroxide have been utilized to control the release of auxins. BNOA is commonly used as an auxin pesticide that regulates plant development through the absorption of roots and leaves, and the biodegradability of BNOA is low, resulting in toxicity and irritation, which can lead to the impaired reproduction, premature birth, and carcinogenity. And BNOA penetrates the wastewater ecosystem via diffusion and presents a serious hazard to human health and aquatic life. As a result, resolving the BNOA contamination of the aquatic environment is top priority. The research objectives are to synthesize the zinc layered hydroxide using self-assembly method, to characterize the prepared zinc layered hydroxide using PSRD, FTIR, and BET surface area analyzer, and to determine the percentage loading of BNOA into the interlamella of zinc layered hydroxide using the CHNS analyzer. Zinc layered hydroxide shares many qualities with the layered double hydroxides, including simplicity of preparation, high biocompatibility, low cost, low cytotoxicity, and complete preservation of the drugs loaded. Zinc layered hydroxide is a mesoporous material that has garnered a significant interest for its ion exchange characteristics and mesoporous structure. Extensive study has been conducted in recent years as the use of zinc layered hydroxide as controlled-release herbicides, medication carriers, flame retardants, and anti-corrosion agents. Next, agricultural technology. Agrochemicals are agricultural-related chemicals, including pesticides, herbicides, and fertilizers. It is frequently used to control pests and boost crop yields in the field. Pesticides are used to increase the crop output and eradicating insects from crops and are frequently utilized in the agricultural area, and herbicides are used to reduce weeds in the agriculture and fisheries. Zinc layered hydroxide is one of the examples of layered metal hydroxide. The most common methods for its synthesis is self-assembly and co-precipitation. The instruments used for the characterization of zinc layered hydroxide is PSRD, FTIR, and BET surface area analyzer. This is the flowchart of the methodology. The first step is to synthesize the zinc layered hydroxide using the self-assembly method, and to characterize the zinc layered hydroxide using the PSRD, FTIR, and the BET surface area analyzer. Next, to prepare the BNOA solution with different concentration, which is the 0.2 molar, 0.4 molar, 0.6 molar, and 0.8 molar, and to intercalate the BNOA using the zinc layered hydroxide, and to determine the quantities of BNOA intercalated. This is the procedure for the synthesis of zinc layered hydroxide. First, 125 ml of 0.5 molar zinc chloride will be titrated with the 2 molar sodium hydroxide under the nitrogen atmosphere until it reached pH 7, and the solution was aged in an all-bath shaker at 70 degrees Celsius for 18 hours. Then it will be centrifuged at 8000 rpm for 5 minutes and rinsed and dried in the oven for 70 degrees Celsius. The sample was grinded and stored in the sample vial for characterization. Next, the intercalation of BNOA. 0.3 gram of zinc layer hydroxide will be added to the 50 ml BNOA of different concentration under the nitrogen atmosphere, and sodium hydroxide will be added into the solution until it reaches pH 7, and the mixture was placed in an all-bar shaker for 24 hours at 70 degrees Celsius and centrifuged 3 times, and placed in the oven at 70 degrees Celsius for 24 hours, grinded and stored in the sample vial for characterization. First, PXRD result. The basis spacing increases when BNOA anion is introduced into the zinc layer hydroxide lamella structure, and the intercalation of BNOA anion into the zinc layer hydroxide interlayer was verified by the observation of a typical intercalation peak at a lower angle, which is 3 degrees and 4 degrees. Intercalation happened as a result of ion exchange process between the chloride ion and the BNOA anion, and the strength of the intercalation peak increases from 0.2 molar to 0.6 molar, indicating the increase of crystallinity. Nevertheless, the strength of the intercalation peak dropped for 0.8 molar BNOA because the high concentration of BNOA in the interlayer, which interfered with the crystal formation during the regeneration. Next, FTIR result. The presence of OH stretching vibration for zinc layer hydroxide at width number 3500 that indicates for hydroxyl group, and the OH stretching vibration for BNOA at 2900 and 2575, the presence of carbonyl group at 1730, the presence of aromatic ring C double bond C at 1600, and the existence of COC stretching at 1220, and the CH2 stretching vibration at 1500, and the stretching vibrations of CO due to BNOA anions at 1100, and confirming the intercalation of BNOA into the zinc layer hydroxide interlamella. The adsorption-desorption for both zinc layer hydroxide and zinc layer intercalated BNOA ZLB is a type 4, which is the mesoporous type material, and the adsorbate uptake was slow, but beyond that, fast adsorption can be detected. The maximum uptake at STP was around 50 cm3 per gram, which indicates that the nitrogen gas was uptake slowly. The adsorption increases quickly at low relative pressure and followed by slow adsorbate uptake at higher relative pressure. Fast absorption can be observed at higher relative pressure, and the maximum uptake was around 9 cm3 per gram, and the desorption branch of the isotope for ZLB was significantly narrower than zinc layer hydroxide, showing that the resultant material has a different pore structure. This might be due to the altered pore structure formed when chloride was replaced by BNOA during the production of ZLB. CHNS analyzer is used to determine the percentage loading of BNOA into the interlamella of zinc layer hydroxide, and it was calculated that the percentage loading of BNOA into the interlamella of zinc layer hydroxide is 83.81%. In conclusion, zinc layer hydroxide was successfully synthesized by using the self-assembly method, and the intercalation of BNOA with zinc layer hydroxide was successfully carried out for all concentration, which is the 0.2 molar, 0.4 molar, 0.6 molar, and 0.8 molar, and the BET surface area decreased from 10.6735 m² per gram to 3.4211 m² per gram, showing that larger molecules of BNOA has been intercalated into the interlamella of zinc layer hydroxide, thus reducing the BET surface area and also the BJH average pore diameter. And lastly, CHNS analyzer shows that the percentage loading of BNOA into the interlamella of zinc layer hydroxide was 83.81%, thus further confirmation that experiment was a success. The first recommendation is more studies need to be carried out to investigate the application of zinc layer hydroxide to intercalate different types of agrochemicals which consist of pesticides, herbicides, and fertilizers. Secondly, layer hydroxides with different metals can be compared with zinc layer hydroxide for the intercalation of each agrochemicals type and investigate different techniques of each synthesis. Thank you so much for listening and have a good day.